G'day, I'm Dr. Philip Button from the Food Entrepreneurship Academy. I'm in the Yarra's Edge precinct in the inner Melbourne suburb on the very edge of the CBD, the suburb of Docklands, where immediately behind me you can see the Yarra's Edge high-rise residential apartments, and in the background on this side here, these are the South Bank high-rise buildings, and then there's the delightful marina here as well. So in this particular video, I'm going to go through the fifth and final video in our series on HACCP and GMP. So I'm going to share my screen and get right into it to finish off the HACCP and GMP video series. So let's start talking about viruses now. In the last video, which was number 4, we spent a lot of time on bacterial hazards. Basically, the whole video was on bacterial hazards. But in this particular video, I'd like to start with, I want to talk about viruses. And remember that viruses are distinctly different from bacteria in that viruses, by and large, don't fulfill the criteria for a living entity. So, they're not actually considered living because, for various reasons like that, they basically don't fit the criteria for life. Whoops. Now, yeah, so, very small particles, of course, they can't be seen without the electron microscope, and don't need food, water, or air because they're not actual living organisms. So they don't cause spoilage, and they infect living cells and are species specific. So what viruses do, they are the ideal obligate parasite where they reproduce inside the host cells, and they need host cells for their survival. So when they're not actually infecting a host, they're in a intermediate stage, if you want to put it that way, an intermediate stage of not actually growing, not actually reproducing, and they're just basically surviving until they can find another host cell or their next host cell where they can take over the metabolic machinery of and make more progeny viruses. But they can survive in the human intestinal area, water or food, for months at a time. And especially in the context of what we're talking about, foodborne viruses, they're typically fecal oral route and related to poor personal hygiene. So remember, we're talking about just in the case, in the context of foodborne viruses. We're not talking about respiratory diseases like COVID-19 here. We're just talking about the foodborne viral hazards. Hepatitis A, for example, is one of those which results in an infection out of the small intestine. There's a range of other, range of symptoms: fever, malaise, jaundice, and a lot of the time they're from cold foods, so foods that are not heated. So we're talking about fruits and vegetables, cold cuts of meats and sandwiches and salads and those sorts of things, where water is used. The water is fecally contaminated, and then that unclean, non-hygienic water is used in the preparation or the cleaning of these particular ready-to-eat products. So therefore, proper hygiene practices, observing, preventing cross-contamination, good sanitation and hygiene are, absolutely essential for this to be prevented. And so we're talking about the food service context in which these are most often therefore transmitted through the fecal oral route. So really, that's all we want to talk about with viruses. And now I'll move on to parasites. So to give you a insight into foodborne parasitology. So parasites are, of course, in the case of what we're talking about, in this case, they are animals. So we're talking about, worms or, worms if they're animals, or protozoan parasites, and they need a host to survive. And thousands, down to the, while thousands exist, only about 100 are known as foodborne contaminants. And as I've already mentioned, parasitic worms, tapeworms, roundworms, flukes, those sorts of things, as well as protozoa are the types of concern, the types of parasites that are of concern from food or water. And again, it's fecal material that tends to be most often implicated in foodborne parasitology. Giardia is one of the most widespread or at least commonly known. And my mind goes back to the late 1990s, where there was a significant waterborne outbreak in Sydney. And the important aspect of this is that its source is animals, so domestic wild animals, and it can go into the water supply. And in fact, with diarrhea, and it's the most common cause of non-bacterial diarrhea in North America. So again, we're talking about contaminated water. Food handlers who have this organism, who are infected, might be asymptomatic or early stages of infection, and these contaminated vegetables that are eaten raw. So, again, just like hepatitis A, it's really important to have good sanitation, hygiene, for food handlers, and effective water treatment as well. Now, Ascaris is a nematode, and it's found in the human intestines, results in an infection, and it has various types of vague digestive tract discomfort, and you can see that transmission is sewage, fertilized fruits and vegetables. So, if that's not treated properly, again, if that's not treated properly, then that's the main issue. And so you can see a bit of a theme here, where it's good sanitation, is important. Poor hygiene, leads to these infections, and therefore, effective, good water treatment is absolutely essential, and education of food handlers to ensure that they are not transmitting the microorganism in the food service context is important as well. So what we want to do now is move on to GMP, actually, after, we last video spent a whole video on bacterial hazards, and this time I just wanted to introduce viruses and parasites, giving two examples, one of an animal worm, one of a protozoan parasite, as as examples of foodborne parasites. But there are others, or waterborne, like Cryptosporidium, Toxoplasma as well. Those are really important. And some, like Toxoplasma gondii, have quite complex life cycles, which go through multiple species as well as humans. So cats in terms of companion animals, and then sheep, for example, as possible agriculture food animals that are implicated in being vehicles of infection that are needed for the transmission to occur. So now, after basically finishing up HACCP and microbiological hazards, we're going to go into GMP and the good manufacturing practice that is important, particularly in the context of dairy product spoilage and basically what happens if that GMP is not observed, then in context of dairy products, what will happen? So to start with, though, it's important to take note that there's short shelf life products such as pasteurized milk, and then there's long shelf life products, UHT milk, cheese, butter, and so on. And the short shelf life products tend to be spoiled through biological or microbial, so microbial growth, bacterial growth, whereas it's enzymes, enzymatic causes which tend to spoil long shelf life products. And it's these long shelf life products which are, some of them, at least with the case of UHT milk, are commercially sterile or considered sterile from a practical point of view, because the conditions actually inhibit microbial growth, and that's why they are, that's why they have such a long shelf life. Psychotrophic Pseudomonas can vary substantially, and it's really related to the conditions in which the milk is produced, or the cattle are actually managed. So the counts are generally low, unless you've got a milking environment or a pasture environment which is particularly conducive for mastitis or some other veterinary medical issue like that. Counts are generally low, but they do increase with low temperature storage of milk, because low temperature storage of milk actually selects for psychotrophic Pseudomonas because of the types of growth temperatures which they do well under, as opposed to mesophilic bacteria. And when they grow under those conditions, they can produce lipase, and these are enzymes which degrade lipids. And these lipases are, tend to be heat stable, even under or after UHT treatment. So there might be substantial UHT inactivation of lipases, but because of the long storage duration of UHT milk, these lipases, even if there's minimal amounts of them there, they can really still act over a long period of time, degrading the fat and causing all kinds of different types of spoilage issues and sensory, change in sensory attributes, off aromas and off flavors, spoiling the product. So this is how it works, especially if you've got damage to the milk fat globule membrane through mechanical means or other means. Then we've got the lipases which can actually enter the fat globule and can affect their lipolytic action on the fat. And therefore, once you've got these free fatty acids being liberated, that's what can actually cause these different types of off aromas and off flavors. So that's why it's really important for good manufacturing practice, right to start from the agricultural environment. So we're talking about pre-farm gate here, to ensure that the microbial count overall is low, and especially the psychotrophic Pseudomonas count is low, and it remains low. So that's why everyday collection of milk compared to every other day collection of raw milk from bulk tanks is, for example, a particularly important strategy for ensuring the good quality dairy products. Not only for liquid milk, but for not only for pasteurized milk, I should say, but also for long life milk and other long life products as well. And these are, this is a little bit on lipase action and the types of sensory defects that do occur. So this lipolysed flavor that can result, which is a pretty general term, but it can be noticeable as rancidity. People just talk about lipolytic rancidity. Anything that just doesn't taste right, basically. And important thing to note is only some free fatty acids do contribute, and they have to be above a certain threshold of concentration to develop those particular lyophilized flavors as well. So, it's important, an important point is that the concentration of individual free fatty acids, so those that are going to result in these flavors, concentration of those is more important than the total concentration of free fatty acids, because, as I pointed above, that not all of them will contribute to this flavor. So you might have a high concentration of free fatty acids, but if the ones that contribute to this flavor are present in only a low concentration, then the milk will still have a good quality taste and aroma. Protease action is quite similar in terms of the proteases that can break down the protein, but in this case we have different types of issues that result: bitterness, bitter flavor from liberation of bitter peptides from proteins, and age gelation are the major defects. So age gelation is where we have protein complexes forming by aggregation of specific proteins, such as casein and beta-lactoglobulin. And it's hydrophobic peptides, which can have a bitter flavor. And if you've got high concentration of those, then you can have the bitterness taking place. So the mean hydrophobicity, must be above a threshold though, and that's based on the amino acid sequence of that particular peptide. Once it's above, a certain threshold, then you'll know that it's got a certain hydrophobicity, which is, likely to result in bitterness. But there's more to the story than that, where there's the structure as well, which can cause bitterness. So we're talking about chemical structure. Now, I just want to finish up after that bit on dairy product spoilage and talk about food preservation by irradiation. And radiation is, it's been used in Japan. I think that was the first place it was used, for like 50 years or something like that. And it's just a fabulous, safe way of, ensuring safe products. So, it can inhibit sprouting, increase shelf life, eliminate insects, and is also really good for food safety as well. So, radiation, gamma radiation, is a really good way of, basically addressing all kinds of food quality and safety issues. So, just to finish up that, it's really absolutely vital for food quality and food safety. HACCP is absolutely essential for food safety, GMP, manufacturing practice, but also good agricultural practice, because I've spoke about the, what can be done on the farm to ensure that there's high quality products. So in terms of hygiene and sanitation on the farm, and like everyday collection of milk from the bulk tank, examples of good agricultural practice. And then there's a whole host of good manufacturing practice that can be applied as well to ensure food safety, maintain the quality of food during the expected shelf life. Okay, so that wraps up our 5-part series on HACCP, GMP, GAP, and it was a really good time to go through these aspects with you. And if there's any further questions, any guidance, support that you want in food safety, food quality, then by all means, please reach out to us or have a look at some of our other videos and any of the other resources that we've got available to support you in the whole area of food manufacturing, from the safety and quality perspective. So thanks very much for listening, and I do hope that you've had a look at our other videos in this series. If not, you can have a look at our playlist here, which contains all the others. And I wish you a very good day. And it's goodbye from the Yarra's Edge precinct in Docklands in Melbourne. Thanks very much, and I'll see you around. Take care. Bye.